Good afternoon. I'm Dr. Brian Shapiro, a Market Segment Manager at ATCC. Thank you for joining us for the latest installment in the ATCC Excellence in Research webinar series. It's entitled CRISPR-engineered reporter cell lines for epithelial-to-mesenchymal transition studies, and it's presented by Diana Douglas and Sangeeta Kumari. Both Diana Douglas and Sangeeta Kumari are experts at using CRISPR-Cas9 genome editing technology for creating advanced cell-based models. In this presentation, our speakers will discuss how this cutting-edge technology was used to create epithelial-mesenchymal transition reporter cell lines. They will then show how these engineered cell lines can be used to monitor EMT and MET via marker expression, small molecule blockade, and migration assays. If you have any questions for our speakers, please use the chat function available through the webinar program. All questions will be answered as time allows at the end of the presentation. The recorded webinar presentation will be archived on the ATCC website, www.atcc.org. So with that, I'd like to welcome our first speaker, Diana Douglas.
So first, a little background on ATCC. ATCC is a nonprofit organization founded in 1925 with headquarters in Manassas, Virginia, and an R&D and Services Center in Gaithersburg, Maryland. It is the world's largest and most diverse biological materials and information resource for microbes. In Gaithersburg, we are an innovative R&D company featuring cell biology, advanced models, and gene editing, which is what today's webinar will focus on. So I'll look at the agenda for today's webinar. We'll go through a brief background on EMT, then we'll look at our vimentin RFP reporter lines, followed by the E-cadherin EMGFP reporter lines, and then conclusions. To introduce today's topic, which is gene-edited EMT reporter lines, I'll start with a brief background on EMT. So epithelial to mesenchymal transition, or EMT, and mesenchymal to epithelial transition, or MET, are involved in many normal and important processes such as implantation, developmental functions like embryogenesis and organogenesis, as well as wound healing, tissue regeneration, and organ fibrosis. So EMT and MET can also become unregulated. This leads to disease and abnormal tissue growth. This unregulated state has been implicated in cancer progression and metastasis. To take a closer look at EMT and cancer metastasis, so during EMT, cells display a progressive loss of epithelial features and gain of mesenchymal features. So rather than it being a binary process involving a complete conversion from the epithelial to mesenchymal or reverse, cells undergoing EMT display an array of dynamic intermediate states, a phenotype that has been referred to as partial EMT. So EMT can be viewed as a continuum. Cells exhibit epithelial, intermediate, or mesenchymal features, and can move through all of these states multiple times during disease progression. When cells begin to undergo EMT in response to an EMT signal, they lose epithelial characteristics, such as the columnar morphology and cell adhesion, and gain more mesenchymal phenotype, such as an elongated morphology, loss of polarity, and increased motility, which allows them to invade. Along with phenotypic changes, there is also a change in the transcription factors and genetic markers they express. Epithelial cells typically express markers such as E-cadherin, ZO1, and cytokeratin, while mesenchymal cells usually express vimentin, NCAD, ZEB1, and SNAIL. So looking at this diagram in more detail, normal cells begin to reactivate EMT, and that allows for the development of the primary tumor site and dissemination to the first metastasis. Often MET is then reactivated, and this leads to the development of the secondary tumor and distal metastasis sites. Because EMT and MET are so heavily implicated in cancer metastasis, and there is still so much to learn about these processes, ATCC sought to provide researchers with gene-edited EMT and MET reporter cell lines that can be used to monitor cellular status changes in real time and as a platform for drug development. One of the novel aspects of our lines is that the commonly used EMT marker genes in our system are tagged with a fluorescent protein to allow real-time tracking of transition. We currently have 6 gene-edited EMT or MET reporter cell lines. So 3 of our lines are tagged with mesenchymal vimentin RFP and 3 with epithelial E-cadherin EMGFP. Our collection includes lung, colorectal, pancreatic, and breast cancer lines, and we will look at all of these lines in more detail throughout the webinar. In addition to the fluorescent tag, another aspect of our line that sets them apart from other models is the editing method. Rather than introducing a transgene that can have confounding effects on gene expression, such as indel creation, disruption of endogenous gene function, or unstable expression of the transgene, we use CRISPR-Cas9 technology for precision editing. In our lines, the knock-in is introduced only at the endogenous locus of the gene, leaving it intact, such that it is expressed from the native promoter. So this allows for a more accurate representation of physiological state. This schematic further details the editing process. This example illustrates the process for one of our vimentin knock-in lines, but the editing process is very similar for all of our lines, for both RFP and EMGFP tagged reporters. So we'll start with the wild type allele from our cell line of interest in panel 1, with the location of the cutting site, which is just before the stop codon in the last exon. Then we introduce Cas9, our guide RNA, and our donor, which is shown in panel 2. Cas9 induces a double-stranded break, so the wild type allele undergoes homologous recombination with the knock-in donor. And then the last panel shows a correctly integrated knock-in allele with the fluorescent tag inserted adjacent to the reporter. After gene editing, all of our reporter lines are thoroughly verified at the genomic, transcription, and translational levels. This slide shows verification at the genomic level. We use junction PCR and Sanger sequencing at the homology arms. So this technique ensures that the knock-in has integrated at the correct locations and without any unintended indel formation. We further verify our edits at the mRNA level through RT-PCR and protein level through co-localization, immunocytochemistry, and off-target analysis. So for mRNA verification, we make RNA and run RT-PCR. Bands are then extracted from an agarose gel and sequenced to confirm the knock-in sequence is correct at the transcript level. For the protein level, we look at intrinsic marker expression along with antibody stain marker expression. So the overlay or co-localization of the fluorescent stains indicates the marker is being expressed at the protein level as well. And we also make sure there is no off-target cutting occurring elsewhere. After we have verified our editing, we use a number of induction or transition assays and biofunctional assays to further characterize our reporter lines. So now we'll take a look at some of the data from these reporter cell lines. The first example we will look at is the HCT116 vimentin RFP EMT line, which is a colorectal cancer model. And the first functional readout we look at in our reporter lines is a change in morphology upon induction, as this is one piece of data that suggests the transition. So HCT116 cells are induced to EMT with a microRNA 200 inhibitor. Minus EMT indicates control cells, while plus EMT indicates cells induced with the inhibitor. And you can see in the image that the cells transition from an epithelial phenotype to a more mesenchymal phenotype. This change is characterized by a loss of tight junctions and spreading out of cells and the loss of polarity. Next, we want to see an increase in intrinsic marker expression upon induction. So this is where the fact that our reporter lines have a fluorescent tag really comes into play. We are able to easily visualize cell transition status in the cells. In this case, we should see an increase in RFP tagged mesenchymal marker vimentin. Again, on the left, we have our control, and on the right, induced cells. So looking at the image, before induction, there is almost no vimentin expression, indicating an epithelial state. Then after induction, we see an 8-fold increase in vimentin RFP expression, indicating a transition to a more mesenchymal status. In addition to looking at intrinsic vimentin RFP expression, we also look for a decrease in an epithelial marker as cells transition from E to M. For the HCT line, we also measured E-cadherin expression upon induction. The image clearly shows a significant decrease in expression and a loss of membrane localization. E-cadherin expression quantification shows an approximately 20-fold decrease upon induction. Another option for quantitative analysis is EMT marker gene expression by ddPCR. Here we synthesize cDNA from induced HCT cells and subjected them to PCR with target-specific primers. The decrease in E-cadherin expression, along with increase in vimentin, ZEB1, and ZEB2, all of which are well-known mesenchymal markers, further indicates a mesenchymal transition. To add versatility to our EMT reporter portfolio, we also have an MET vimentin RFP reporter line and the MDA MB231 vimentin RFP line, which is a breast cancer model. Cells are induced to MET with U0126, which is a highly selective MAPK inhibitor. And literature has reported this is an effective means of inducing MET transition in MDA cells. So here you can see the mesenchymal to epithelial morphology change as cells transition from spread out cells lacking polarity on the left to more tightly packed cells with columnar morphology on the right. In these cells, we are looking for a decrease in endogenous vimentin since cells are transitioning away from mesenchymal to epithelial. So this image shows that induced cells display a significant decrease in vimentin expression upon MET. In addition to the induction and transition assays we conduct on all of the reporter lines, we also look at biofunctional characterization. These next two slides show examples of biofunctional data from our A549 lung cancer vimentin RFP EMT line. The first piece of biofunctional data we look at is invasion. Invasion is a key characteristic of EMT transition. As cells become more mesenchymal, their invasive capability should increase, which helps them disseminate and metastasize. So in this line, EMT is induced by TGF-beta for 3 days. We then measured invasion using a transwell invasion assay, and in the image, examples of invading cells are marked with red arrows. So for A549, we can see about a 3-fold increase in invasion upon EMT induction. Another exciting piece of biofunctional data comes from drug testing. For the A549 reporter line, we used 2 small molecule EMT inhibitors to block transition. 2 pathways associated with EMT were targeted, TGF-beta and SRC, using A8301 and PP1, respectively. A8301 is a TGF-beta kinase activin receptor-like kinase inhibitor that blocks phosphorylation of SMAD2 and inhibits TGF-beta-induced epithelial-to-mesenchymal transition. PP1 is a potent and selective SRC family protein tyrosine kinase inhibitor. So SRC has been shown to play a role in E-cadherin regulation and epithelial-to-mesenchymal transition. Increased SRC activity promotes EMT, while SRC inhibition suppresses it. So on day 0, A549 vimentin RFP cells were seeded with and without stem ex vivo EMT induction supplement, along with the respective DMSO or drug dose. RFP expression was monitored for 5 days and then quantified and plotted on day 5. Both inhibitors were effective in blocking EMT. A8301 shows a dose-dependent reduction in EMT, as evidenced by the reduction in RFP fluorescence intensity. At the highest dose, EMT is almost completely blocked. PP1 also displays a dose-dependent decrease in EMT. So this data highlights the opportunities these models present in the field of drug development and drug testing. As some researchers prefer to study E-cadherin as their marker gene of interest, we also have 3 E-cadherin EMGFP lines. The first ECAD EMGFP line we will look at is the pancreatic cancer model PANC1 ECAD EMGFP MET. So as with our vimentin RFP reporters, the first thing we look at is morphology change upon induction. In this case, we are able to induce MET with microRNA 200 treatment, and we see a transition from a mesenchymal phenotype as seen in the top panels to a more epithelial phenotype seen in the bottom panels. In terms of intrinsic reporter expression, we are looking at epithelial marker E-cadherin now. So in the PANC1 line, we see a significant increase in ECAD expression as cells transition from mesenchymal to epithelial. So we quantified this increase to be greater than 2-fold. Our second marker in the ECAD EMGFP lines is a mesenchymal marker. For this reporter line, we use SNAIL. SNAIL is a transcription factor that promotes the repression of E-cadherin. So you can see that upon MET induction, SNAIL expression decreases by about 60%, further indicating a transition away from a mesenchymal phenotype to an epithelial phenotype. Biofunctional data for this line shows a decrease in the cell's invasive capabilities upon induction to MET. Using a transwell invasion assay, we found an approximately 6-fold decrease in average number of invasive cells. We have two additional ECAD EMGFP lines that will soon be available. The first of which is the BT474 breast cancer ECAD EMGFP EMT line. Again, the addition of this line adds variety to the portfolio by providing an ECAD EMGFP EMT model in contrast to the PANC1 MET line. In this line, cells can be induced to EMT with stem ex vivo EMT induction supplement. You can see the minus EMT control cells on the left are very epithelial in nature and then adopt a much more mesenchymal phenotype upon EMT induction. The BT474 ECAD EMGFP EMT reporter cell line also displays a very obvious intrinsic marker change. The E-cadherin expression decreases significantly, about threefold, as cells are induced to EMT transition.
Well, thank you, Diane. Now we'll go ahead and transition over to Sangeeta, who will discuss ATCC's latest EMT reporter model, MCF10A E-cadherin. Sangeeta?
Thank you for a nice introduction. Now let's focus on our breast epithelial MCF10A EMT reporter line. To create MCF10A EMT model, we targeted an epithelial marker gene E-cadherin to tag with reporter EMGFP and used CRISPR-Cas9 technology to insert reporter sequence at the target locus in the cell's genome. The advantage of using CRISPR method is that it allows the reporter sequence insertion at the endogenous locus of the gene such that it is expressed from the native promoter, which allow accurate representation of physiological state. And also this method prevents unintended mutation, disruption of endogenous gene function, or unstable expression of transgene. This diagram here illustrates the process of gene editing to generate E-cadherin knock-in lines, and the editing process is very similar for all of our other EMT-MET reporter lines. For MCF10A EMT line, we started with the wild-type E-cadherin allele with the location of the cutting site just before the stop codon in the last exon. Then we introduced Cas9 guide RNA and our donor, which is shown in panel 2. Cas9 protein induces a double-strand break, so the wild-type allele undergoes homologous recombination with the knock-in donor. And the last panel shows here a correct integrated E-cadherin GFP knock-in allele. After gene editing, our reporter lines are thoroughly verified at the genomic, transcription, and translational level. For genomic level confirmation, we use left and right junction PCR and Sanger sequencing to ensure that the knock-in has integrated at the correct location and without unintended indel formation. Left junction of knock-in confirmation is shown here. PCR was done using primer pair located at GSC region and outside the left homology arm. The DNA gel shows the expected amplicon size about 0.8 kb in the knock-in cells and no amplicon in the wild-type or parental line. Sanger sequencing of PCR product further confirmed the peptide linker and EMGFP knock-in sequence just before the stop codon in exon 16 as expected. This is again PCR data confirming right junction of the knock-in. Primer pair used are located in GSC sequence and outside the right homology arm sequence. DNA gel showed presence of 0.6 kb PCR product in the knock-in cells and no PCR product in wild type. Sanger sequencing of the PCR product confirmed EMGFP sequence in exon 16 before the stop codon in the cell's genome. Following genomic level confirmation, RT-PCR and Sanger sequencing are used to confirm fusion E-cadherin GFP transcript. RT-PCR was performed using primer pair located on exon 15 and 3' UTR region between the knock-in sequence for both wild-type and gene-edited cells. DNA gel indicated the presence of 479 base pair in wild-type, indicating no knock-in sequence and 1214 base pair in the knock-in cells as expected. Standard sequencing of PCR products showed the peptide linker sequence followed by EMGFP sequence just before the stop codon in the knock-in cells, confirming the correct integration of EMGFP in the cell's genome at the transcription level. To confirm the presence of E-cadherin EMGFP fusion protein expression in the MCF10A EMT line, we performed immunocytochemistry. Gene-edited reporter cells stained with anti-E-cadherin, shown here in red, and endogenous E-cadherin EMGFP expression in green. Merged red and green image in the right confirms the co-localization of the E-cadherin EMGFP and stained E-cadherin protein, indicating that the E-cadherin GFP fusion protein is being expressed at the protein level in the reporter cell line. After validating the integration and expression of E-cadherin GFP in the MCF10A reporter cells, we wanted to make sure gene-edited reporter cells and parental cells are similar in terms of morphology and growth. Here I'm showing the phase image for parental on the left side and gene-edited MCF10A EMT on the right side. Both cells showed the tight junction epithelial-like feature and looked very similar. Same growth medium was used to culture both lines. Growth rate and pattern, parental and gene-edited, were also very similar. Population doubling time for parental was 42 hours and for gene-edited 45 hours. Difference in growth was in acceptable range. First step to confirm successful EMT cell line model is to show characteristic morphology change as cells transition from epithelial to mesenchymal stage. To show MCF10A EMT cells undergo successful EMT, cells were incubated in complete growth media, supplemented with EMT-inducing supplement, or an equivalent volume of PBS as a no treatment control for 5 days. We observed the morphology of MCF10A EMT cell line changed from epithelial-like tight junction, closely packed, a cobblestone appearance to mesenchymal cells of a spindle-like shape, less packed upon EMT induction. This morphology change demonstrated that MCF10A EMT reporter cells are able to transition successfully from epithelial to mesenchymal stage after EMT induction. As cells transition from epithelial to mesenchymal state, epithelial marker E-cadherin expression decreases. Therefore, for function data to support our product, we first demonstrated a significant decrease in E-cadherin GFP expression in MCF10A reporter cells after EMT induction. Here, top panel includes DAPI and GFP image for control uninduced reporter cells, and bottom panel for EMT-induced reporter cells. EMT-induced cells shows a significant decrease in GFP expression compared to control, and upon quantification, we found there was greater than two-fold decrease in GFP expression upon EMT induction. A successful EMT induction causes decrease in epithelial marker expression and increase in mesenchymal markers expression. Therefore, we also looked at two mesenchymal marker gene expression, vimentin and fibronectin, in MCF10A EMT upon EMT induction. Top panel is ICC stained fluorescence image for vimentin marker in control and EMT-induced MCF10A reporter cells, showing increase in vimentin expression in induced cells. And bottom panel is showing increase in fibronectin expression in induced cells. Both markers increased by more than 50% upon EMT induction, as shown here in the column graph, supporting a successful EMT induction in our MCF10A EMT line. For the final function data, we tested the migration ability of EMT-induced MCF10A EMT cells. Invasion or migration is one of the key indicators of epithelial to mesenchymal transition, as mesenchymal cells are considered more able to migrate and invade, which helps them disseminate to establish metastasis. We performed a scratch assay to assess migration capacity of EMT uninduced and induced cells. A scratch was made after 6 days of EMT induction on a confluent monolayer, and images were taken at 0, 16, and 24 hours after scratching. Top panel are control cells that did not go through EMT induction, and lower panel is EMT-induced mesenchymal cells. EMT-induced cells displayed a significant increase in motility as the induced cells moving to gradually close the scratch or wound by the end of 24 hours compared to control cells. In addition to scratch assay, we also performed transwell migration assay by seeding cells on the upper layer of cell culture insert with permeable membrane. Following 24 hours of incubation period, the cells that have migrated through the membrane are stained and counted. Here, migrated cells through membrane are marked by red arrows in the top panel phase image for both control and EMT-induced conditions. We observed more number of EMT-induced cells migrated through the membrane compared to control non-induced cells. We quantified the number of migrated cells and found about 10-fold increase in migration upon EMT induction, as shown here in the column graph. Scratch and migration assay both demonstrated increase in motility and migration of MCF10A reporter cell line upon EMT induction.
Well, thank you, Sangeeta. Now that Sangeeta has finished her presentation on the MCF10A E-cadherin model, I'll pass the microphone back over to Diane for summary and conclusions.
So in conclusion, all this data gives an overview of the EMT and MET reporter cell lines created by ATCC scientists using CRISPR-Cas9 editing. Through the use of precision editing technology, we have created reporter lines that have a fluorescent tag on the reporter gene while leaving the endogenous locus intact. All of the reporter lines have been thoroughly validated at the genomic, transcription, and translational levels. And following verification, EMT and MET reporter lines were further characterized with induction or transition assays, as well as biofunctional analysis. So these lines provide a novel tool for researchers to monitor cellular status changes in real time, to further understand the mechanism behind EMT and MET, and as a platform for drug discovery. For more information about any of our EMT or MET lines, please visit the website at atcc.org/EMT. And I just want to say thank you to the R&D department here in Gaithersburg and to everyone listening, and we look forward to your feedback and questions.
Well, thank you, Diana. In just a few moments, we will begin our Q&A session. Please use the chat function available through the webinar program to submit your questions. The recorded webinar presentation will be available on demand on the ATCC website at www.atcc.org. Sangeeta had to step out of the room for a moment. However, Diana can certainly take your questions, and Dr. Utsav Sharma, who is ATCC's new product manager for oncology products, has joined in. So I guess I'll go ahead and start out with the first question, which I'll take, and that's, will the webinar recording be available to view later? Which is, the answer is yes. The recording will be on our website by Monday. You can find it at www.atcc.org/webinars. Simply navigate to the year 2023, and you can find the webinar. All right, let's go ahead and jump into some questions around the data or around the presentation. So, first of all, Diana, can you describe the cell invasion method that we used?
Sure. So we used a transwell invasion method where the cells are seeded and then induced for about 3 to 5 days. Then they're transferred to invasion chambers where the invading cells have to transfer through a porous insert with a matrigel coating to the chemoattractant below in the well.
Okay, good, good. Here's an interesting question, and it's about the CRISPR gene editing part of the product generation. Do the EMT reporter cell lines have any undesirable mutations or indels caused by the CRISPR editing process?
So we did actually do analysis on 10 of the most probable potential off-target sites for our guide RNAs that we used to create the reporter knock-ins. We then amplified those off-target sites and sequenced all of them, and we did not see any indel formation on those off-target sites that could have been caused by CRISPR-Cas9 editing.
Okay, good, good. And do you know if the cell lines have a single allele knock-in or double allele knock-ins?
So based on our RT-PCR data analysis, the knock-in does seem to be homozygous.
All right, good, good. And just out of curiosity, do you know if the E-cadherin or vimentin expression in the knock-in cells is similar to the parental?
It is. We we did a Western blot to confirm the knock-in. We also did ICC colocalization.
Right, right. The immunocytochemistry that you showed, right?
Yes, yes.
Okay, cool. And this next question is just about products that we might be working on in the future. Do we plan on making one around melanoma or maybe for blood cancer research?
We don't have those models, unfortunately, available at this time, but it is a possibility in the future, and I can definitely pass on that, we do have customers interested in those types of lines.
Okay. And this next question that's come in, it is around the microRNAs that we use to induce EMT. So you showed using one. Are there different miRNAs that you could use?
Yeah, so the microRNA 200, that's the most common one used in EMT studies, and that's the one that we used for HCT, like you said. But there are others that are sometimes used depending on the cell line. For example, I've seen EMT promoters like microRNA 155, 27, and 9 used. It just depends on the cell line.
Gotcha, gotcha. Okay, and what about using something like TGF-beta1? Could you use that to induce EMT?
You could use TGF-beta. There's also like R&D Systems has a stim ex vivo EMT inducer that we've used. There are several different ways to induce EMT in a lot of these lines.
Nice, nice. Now, you showed us a lot of data in this presentation. Is this published anywhere aside from ATCC's website?
Sure. Yes, we do have one recent publication. We co-authored a paper with a group from the University of Pittsburgh, Dr. Zing's lab. The article was in the September 2020 issue of Science Advances. So they developed a live cell imaging platform to track EMT in real time using our A549 line. So the title of the article is Live Cell Imaging and Analysis Reveals Cell Phenotypic Transition Dynamics Inherently Missing in Snapshot Data. So that is available on PubMed.
Excellent, excellent. I know you must be proud of that. Well, we all are.
Yes, that was a great collaboration with Dr. Zing's group there.
All right. Now, could you talk a little bit about the applications for the EMT cell lines?
Sure. So there are several, possible applications. Some groups used it for high throughput discovery of new anti-EMT drugs. It can also be used for tracking of EMT and MET cell status in real time, like I talked about with the publication, by monitoring the RFP or the EMGFP expression. They can also be used to elucidate cell signaling pathways involved in cancer progression.
Okay, good. Now, this next question is around media requirements for the cells. Is there any difference between the cell culture medium or method used for the EMT line versus the parental line?
No, we use the same media and the same, culture method recommended by ATCC for both.
Okay, and kind of along the same line of questioning, did you notice any differences between the reporter lines and the parental cells in terms of growth rate, morphology, and other physiology functions?
They were all very similar. That's one thing that we looked at for each of the lines to make sure the starting state of the cells was normal.
Okay. And let's see. The question came in directly about the MCF10A reporter line, but really this could be directed towards any of the EMT, MET reporter lines. Do they contain any antibiotic gene sequences at their, either E-cadherin or vimentin loci?
No, so we use the precision gene editing and homologous recombination. So it allows for really precise editing of the cell line without disrupting the endogenous locus.
Gotcha. We had a question, a couple of questions come in that were very similar, and maybe you could answer them both sort of in the same sort of frame. So one, what is the peak time of expression after induction? And then what are the half-lives of RFP and GFP in the reporter cell line?
So the peak time of expression after induction is typically 3 to 5 days. It varies a little based on the cell line, but average is about 3 to 5 days. I don't know specifically the half-life. I never had an issue where I in my analysis where I saw like degradation, or had a problem with the signal lasting long enough to do a complete analysis. That's something I could look into. I'm not exactly sure exactly what the half-life is.
Okay. No, that's a perfectly good answer. So in your hands, that just wasn't an issue.
Right.
Okay, good. So are these cell lines clonal?
So they're from single cells.
Okay. So single cell clones.
Yes.
Gotcha. All right, let's see. This next question, great talk. They missed the front part of the presentation. Did we use flow cytometry to select the E-cadherin GFP express cells?
We didn't
use in the analysis or in the development of the cell line? I'm not sure.
I'm going to say in the development, did you use flow?
For some of the lines, we use like a Sony cell sorter to select our single clones, just depending on, you know, how many positive clones we were able to get. Sometimes in, you know, the editing process, you're not getting a ton of clones on their own, so you do, you know, we would sort. It depended on the line, but sometimes we would sort for the fluorescence. Yes.
Okay. No, that totally makes sense. That totally makes sense. So we have one more question that's come in. So if you guys have any questions, please feel free to drop them in the chat. So I'll go ahead ask this. We'll wait a moment. I'll have the final word, and then we'll close.
Okay.
So Diana, did you use these EMT cell lines to set up some mouse model? And you know, this is specific. This is rather vague. So any animal model.
So we didn't do any animal models. No, we didn't do any in vivo work. All of our work is in vitro. I know some of our customers and collaborators have used these lines in vivo, and we, you know, it's great to see if customers have data that, you know, to share from, you know, mouse models or in vivo work. But in our development, we do strictly in vitro.
All right. Sounds good. Sounds good. Okay, well, it looks like all the questions have come in. So while we wait just to see if any more, I'll just say a quick message. Coming soon, we have a webinar next week presented by Steve Budd and James Clinton on optimizing your 3D culture workflow with organoid growth kits. That's going to be at 12 noon on June 29th. All righty. Well, I guess at this time we can conclude our Q&A session. I'd like to thank Diana and Sangeeta for the excellent presentation, and thank you everyone for attending this webinar. Thanks again for your time today, and have a great day.